In this section, we're looking at the gene detection motivation, and now turn it into the computer science problem of computing how much signal we get from specific open reading frames in particular. And one key question for this is how big genes really are. If they were all very, very short, or if they were all at least in a specific range, we would be very much out of luck. It turns out that they are usually much longer than is needed for this. The specific gene in the genome browser that I've shown you briefly, and I'll go there in a second just to show it again, is this MC1R gene, which is one of the key genes for red hair, coding for the specific type of red hair, often combined with bright skin, etc. So it's one of the genes where you can directly see if a person carries it or not. So it's a bit of a rare event or a rarity in humans that the single gene is so visible. Therefore, there's even a magazine called MC1R magazine, maybe with specific, I don't know what articles they have. There was one about sunscreen. Let me briefly show the genome browser again. That's this website where you can zoom in on any part of the human reference genome. And they have essentially collected a lot of information that is known or suspected about specific regions in the genome. And here's the MC1R gene specifically. It starts at precisely this point with the start codon, as you'd expect. And then it keeps going for a very long time. Here's the sequence in the DNA, and here's the translation to amino acids, 3 at a time, exactly as in the video we've seen before. And you already see you can keep going for a good bit. And I'll zoom out a bit here to make it more visible. So you can already no longer see the DNA sequence. You can still read the amino acid sequence. If you go further out, then even that is subtracted into some just some bar. And okay. Maybe we'll have to leave it with that. I don't want to keep waiting for the website for the rest of our lecture. So there is an end to this gene. That's as much as I will promise. Okay. And if you go to the end, there is a stop codon. Maybe I'll leave it with that, and you can check for yourself if you want. A few basic facts just about the human genome. Only roughly 10% of the entire 3 billion bases are known genes, I guess we should say. Is around, I didn't write down the number. Oh gosh, it's around 20,000 genes, roughly. There was a suspicion at the beginning of the Human Genome Project that it would be 5 times as much, and people were a bit astonished at the time that the number of genes seems to be much lower. Anyways, what's the length of the genes? That will become important for us in a second. There are some genes that are very, very short, just 63 bases. This BP is the usual unit that people give. It's just letters. People write BP because it's base pairs. And you'll remember that DNA always has the double strand. This also means, by the way, that typically genes can be on either strand. The translation doesn't matter. So you could either have the gene in the forward direction, the way we've seen it in the genome browser, or it could be the reverse complement, where every letter is replaced with its partner, and it's reversed. So whenever you do something with genomic sequences, you have to keep that in mind, that there might be both options, and either one would work. So this one's an exception. The otherwise shortest protein coding genes were more on the order of a few hundred bases. The longest one that is known as 2 million bases. That would be an enormous protein, but it turns out almost all of this is actually cut out during translation. So there's these sequences that are called introns. They're part of the gene in the DNA sequence, but they will never end up in the protein because before the messenger RNA leaves the nucleus, there's loops of this cut out and connected again. The video just showed a segment of this RNA vanishing. The way it's really happening seems to be there's certain parts brought together, then there's a loop formed, the loop is cut out and recycled, and the rest is glued together. And in this case, for this enormous gene, 99% of this long sequence is cut out again. Why things are done this way, it's not 100% clear. It might just be a way to slow down the translation of this gene, because this guy is so long, it's taking roughly 6 hours to produce one copy, just because the copying process takes a certain amount of time. I think I read maybe 2,000 base pairs per minute is the translation speed. It's not super fast, but it's happening at massive parallel scale, right? All the time, 24/7, and in all the cells. Okay, so a typical length of a protein coding gene is of the order of a few thousand base pairs. That's what, from this long story, that's what you should keep in mind. The rest ignore, okay? A few thousand base pairs. Is that good or bad news? Can we reliably distinguish these from just having some random open reading frames? I want to ask you this question, but let me define the model first. This is the null model. Suppose there was no gene there. We pretend we found some open reading frame. Now we want to know, is this an actual gene or am I just seeing a statistical fluke? Is this, by random chance, there's a start and a stop codon and the whole thing in between means exactly nothing? How can I distinguish these? We'll analyze the probability that this gene forms in a random sequence that's completely without any meaning. So we pick a long DNA sequence. Every character is uniformly at random, independently of all the others, chosen from the 4 options. And then the question is, if I have some occurrence of the start codon and then the next occurrence of the stop codon, how much is in between? That would be an open reading frame. And that's what I'm comparing with the one I find in the the genomic sequence. And so let's play this game. I mean, I haven't told you, so there's no right or wrong answers at this point. What do you think? Just intuitively. Type it on on Slido if you can. Then we can record it also for the people in the live stream. And here I mean 20,000, that was in the actual human genome. And we want to compare this with a random DNA, okay? So you're just generating your rolling dice instead of using an actual genome. Probably what comes out is not it's not capable of living. It's a probabilities question, not a biology question. Well, since this is not a knowledge question, let me, okay, maybe I don't want to influence, if other people are still thinking, maybe it's best to get unfiltered, what do you think individually, and then we'll do the comparison afterwards. And I get to enjoy to see already what you voted. Anyone still thinking or still typing? So let me show what we've got so far. 20,000 and in the order of millions. And let's do a quick back of the envelope calculation first. And I'm already gonna tell you that this is not an accurate way to calculate things, and you can try to think about why, and then we'll do the proper solution. But I think this is insightful to do. I guess I should also draw an envelope, right? But it's the back of the envelope, so you can't really see the front. Okay, I'm not an artist. So we have a sequence of DNA which is not dotted. And suppose there's the start codon. I'm really embarrassing myself. I don't want to mess it up. What's the ATG? And let's pick TAA. I can remember that one. That's what this thing has to look like. And in between, no stop codons, right? In all of this range. That's the important bit. Now the start one, we're only start looking once this has happened. So I'm assuming we're sitting right here. And now the question is, when do I see the next stop codon? Now let's do something simple. What's the probability of any specific 3-letter sequence being a stop codon? So what's the probability that if you take 3 random letters, it's one of the stop codons, TAA, TGA, and I'm really not, TAG, TGA, both. Goodie. Say it again. That's right. This is 3 out of the 64 options of picking 3 letters in this model. Is that big or small? Well, who knows? I mean, it's a certain number. It's roughly 1/32 or so, a bit more, right? That would be 2. Maybe it's 1/20. So I don't know. Very, very roughly, something like this. What does that mean? Well, it means whenever you move here, you have a 1 in 20-ish chance that the next thing stops the sequence. So in expectation, maybe we should expect 20 or so of these codons, and maybe then roughly 60 as a ballpark for the length until we see another stop codon. So maybe around 20 codons. Another way to say it is if you look at the codons, out of 64 codons, 3 are stop. So that's the same thing again. So 60 base pairs. Again, this is incorrect in a specific way, but is also the order of magnitude is not wrong. If you compare that to what we've had, 64 base pairs for this smallest degenerate one, but most genes of the order of a few thousand, that's great, because that means if you find this very long open reading frame, you can be quite sure, intuitively or qualitatively, we're quite sure it's an actual gene, because if the whole thing was random, it's very unlikely to see such a long reading frame without a stop codon all the way. Now let's get a bit more precise. because there's some cute math that you can apply to this, and it's with these kind of probabilities, you can, in hindsight, I can say what we got with the back of the envelope method is the right ballpark, but you don't always know up front. So what we want to answer this question a bit more accurately is the expected length, say, of a word that we assume we've just seen the start codon, so we're starting right after this. Now we count how many letters do we see until we, for the first time, see one of the stop codons. I apologize, I've used the U here again because we're in RNA world. It's the same, right? Just replace T and U whenever you see them. Maybe we should decide for one letter and just ignore it. So we've just seen the start codon. Now we keep reading letters until for the very first time we see one of the stop codons, and then we stop. That's a language in the formal language sense of all strings that end with a stop codon and do not contain a stop codon anywhere before that. Now it turns out you can write this question as a regular expression, or you can draw a DFA, a deterministic finite automaton that accepts this language. In this case, it's relatively straightforward. So I've just done the example, and we, in a way, will revisit this question and how to systematically compute this in general in one of the upcoming units. The states in this DFA will just encode the last letters we've seen, and more precisely, the longest prefix of any of the start codons that we've seen so far. So we start with having not seen anything, the empty string. If we see a U, which is the first symbol of all the three start codons, then we make progress and we remember we've seen a U. Now, along the bold lines, we make progress in the same way. We can continue reading one of the stop codons. For example, UGA brings us here. Then these accepting states mean we stop. But if I'm, say, in this state, I've seen a UG. If now an A comes, I know what to do. But if I see a U instead, I can't continue. I have to give up. This UG prefix is not going to continue. But I have to jump back to this state because the U I'm reading now might be the beginning of a new occurrence, of a potential occurrence. Was there a question?
The sequence doesn't matter, like do we get like U, or because stop codon is AUG, so should not we get A first and then U and then G?
We need to see them in the right order, that's right.
UGA is also the final state which is affecting this, but we want AUG.
No, AUG is the start codon. And this one is not part of the automaton. That's why it's the same as in this picture. I'm assuming we've just seen the start codon. And now we wait for one of the stop codons, which are these three. Okay, it's just by chance that one is the reverse of the other. So what confused you, I think, is that AUG is the start codon. It's not exactly this reverse, right? UGA is a palindrome, but I guess there's so few letters, that's not so surprising. So AUG doesn't occur in the automaton because we assume we start the game here once we've seen the start codon. We're only interested in the conditional, so the conditional number of characters you still have to read after seeing the start codon before you see the first stop codon. And that's the language that this automaton accepts. It can be described this way. You can, of course, from the automaton read off other descriptions of the language, but it turns out the automaton is the most natural one. And again, just to reiterate, is there any other interesting? In this case, it's relatively easy. There's only this one case. There's sometimes a state like this UG state, where if you read a U now, so here, this state means we've seen some string and it ended with UG. And now there's a new letter coming, maybe a U. what's the best guess? What's the longest prefix of any, and longest beginning of any stop codon that I could have seen at this point? And that's only U. U might be the first letter of any of these. The UG are kind of not going to lead anywhere, so I'm forgetting them. Is the automaton kind of clear? To make the connection for people, this is essentially the Aho-Korosik automaton or string matching automaton for a single string. I'll explain these in more detail in a later section in the kind of systematic way. Here for these specific strings, it's kind of easy enough that we just manually figure out what this automaton should look like. Okay, from this we can analyze how long the expected open reading frame in a random string would be precisely.
The C and G should be separate. That's step from UG to backward.
This one?
No, CG.
C and G?
C and G should be in separate string.
Yeah, so what I mean by this kind of a bit of a sloppy shorthand notation, I guess, it means any of these letters. The automaton always reads a single letter. And here C and G do the same thing. They both go back to the start state, same as here. Anything that's not a U keeps you in this initial state. Yeah, but that's important. That's a good point. Okay, so what we'll do is analyze the expected number of characters from a given state to some accepting state. It's one of these cases where it's easier to solve a more general problem. We're actually just interested in the number of steps it takes from the start state to any of these accepting ones. We'll do it for all the states, because then you can relate them to each other and calculate it for all. That turns you, in particular, it returns the answer that we want for the state Q epsilon. So I'm just gonna, here maybe I should have called them Q subscript, but that's less readable. I want to make clear here that I'm talking about the state and not just the string epsilon. We want this for Q epsilon, but we'll do it for all of them. For some of them, that's really easy. Let's call this number, let's call it T, and then this substring X. That's the expected number of characters from the state where we have this label X to some accepting state. I'm just going to be a bit sloppy with the notation, but I could have listed all these three. So we'll need the automaton to keep things in mind. Okay, my laptop claims it's lost connectivity. That's not good for the live stream, but we're gonna ignore that now. So this is the TX. That's what we're gonna write down. There's some of the states where this is super easy, namely all of the stop codons. I'm already at the state, so there's nothing more to expect, right? It's just 0. I'm done there. For all the other states, we don't know exactly because it's random. The next letter is random. But we're talking about the expected value, so we can still say something. For example, for UA, there's a 1 in 4 chance to go to TUAA. And there's another 1 in 4 chance to go to TUAG. Now, of course, I could have shortcutted this because these are contributing 0. But there's one thing missing. I'll come to that in a second. Then there's also these 2 outgoing edges for U and C. So with probability 1/4, I'm going back to TU. And with probability 1/4, I'm going back to T epsilon. But I shouldn't forget that I've taken a step. So I also need to add +1. In all of the cases, I'm making this one character that I'm reading now. Okay? And maybe it's kind of obvious already from this example, you can do this for all the states. For all the states, you can sum over the different outgoing steps. And you're going to arrive at some system of equations. Let me do one more just to make it clear. I'm looking at this state. There's a 1 in 4 chance of going here. There's a 1 in 4 chance of going back to TU. And there's a 1 in 2 chance to go back to T epsilon this time. And I'm always doing my one step. Yep.
The second step, there are only 3 possibilities. So why is like write 1/4 and then other 1/2? I think it should be 1/3, right?
Which one?
The second TUG one.
This one?
Yeah. Should it be
This is 1/4. Yeah, you're right. Yeah. I maybe said the right thing, but I wrote the wrong thing for sure.
But still, I mean, I'm saying that it's only 3 possibilities. So it shouldn't
Yeah.
be 1/3.
They're not equally. No, look, these are 2 different letters. So there's always 4 different outcomes, but 2 of them do the same thing. So this arc is much more likely to be taken. Because we're doing one step. We read one letter, so we have to count that letter. Otherwise, if you leave out the +1s, this is all going to solve to 0, right? If you finish the set of equations. Okay, so we could write this all down, and we could go back to middle school and solve the system of linear equations. Boring. So I prepared this a bit. And I thought it's about time to also show some tools, given that the module inadvertently is classified as a practical module. Maybe that's even more fitting. So I coded this up again in Mathematica, and let me get some AI help to translate it to SymPy. But it's really nice to write. So I encourage you strongly to kind of learn these tools instead of doing stuff manually in this case. So you can define your variables. These are exactly as I had them. They're having the same meaning, right? And there's a cute way to do this with just from one string. That's very concise notation. I mean, in SymPy, symbols always have a string representation. This is just cosmetics for us humans. You wouldn't need that for the math. And then we write down equations. Now, here's a bit of a nuisance in SymPy. If you want an equation A equals B, you have to write it as something equals 0. And then you only give the left-hand side of that. So let's pick an example that we've done. Well, the easiest ones are all the stop codon parts. They were just equal to 0. So here in this list of equations, they just show up as a term. Is this large enough to read? Ish. Yeah, it's not showing. Maybe just here the browser zoom. We've also looked at this one, the TUG. This was the bottom one where we had the two. There was this one option of going to the accepting state, and there was the 1 in 4 chance of going back to TU, and the 1 in 2 chance. That's 2 times. It's a bit silly way to write, but okay. Going back to the start state, T epsilon. Okay, so that's all I've done, and I've done this for the other states, but this is very superficially just read off from the automaton. It tells you exactly what to do. Then you get these equations. You have to define the set of variables again. And then there's a function you can call to solve them. So I might need some time to set up the virtual machine. But then the solution you get is essentially a map for all the symbols, and we care about t epsilon 1 from the start state. How many steps do I need? Then you can get a symbolic solution. Here it is. This is the map, right? You see from t, it takes 64 third steps in expectation. It's not that much, right? Remember, we had on the order of 60 from the back of the envelope calculation. In reality, it's more 21 1/3. And you can compare this with maybe some naive estimate if you just, I guess this is a slightly different well, if you do the math maybe right. That's another back of the envelope calculation. And this value is much smaller. That's the one thing we can take from this straight away. Okay, now you know how to solve linear equation with a big sledgehammer of a tool. Maybe that's not needed in this case. You would have been able to solve it manually. It's not that hard. The key insight here was we can write down this automaton for these strings. You have to work a bit to get the transitions right, so that you always take a transition to the longest potential prefix of one of the codons that exist. But in this case, because they all start with a U and there's no other U in them, the process is fairly simple. Okay. So the biological message is it was 64 thirds, or it was 21. Let me cheat again. Don't want to. Yeah, 64 thirds. Well, that's the way I learned to write it, with periodic fractions. So a good deal less than the 60 that we estimated from the back of the envelope calculation. Again, in this case, not a big deal because the genes are orders of magnitudes longer, but also we're half an order of magnitude off of our intuitive guess. So one wants to be a bit careful with these calculations.